Great. Hi, everyone. Welcome to our Clinical Trials Community of Practice seminar. My name is Nasia Safdar. I'm the faculty lead for this seminar series. Our speaker for today is our own Dr. Mike Semanik. But before we go to his talk, we just have an announcement with a couple slides from a group from Albert Einstein School of Medicine on disability as diversity, reducing researcher roadblocks. So we have Professor Karen Bonnock here and Patrick George, and I will turn it over to them.
Thank you so much. I just wanted to reach out and let you know about we call it D2 or 3 study, which we'll tell you about. We're only take a couple of minutes because there's an opportunity for all of those on this call and probably many of those in your lab or collaborators to participate in this part of the study, which is a survey, and it's either be approved and you'll be reimbursed or have the option to donate to a nonprofit. So can you please do the next slide? Thank you. Okay, so this is our element E study for our Einstein-Montefiore ICTR. And the idea is based upon the fact that given the relatively high prevalence of people, particularly adults with developmental disabilities, I don't want to, I don't have time to get into the definition of those, but they roughly encompass Down syndrome, autism, cerebral palsy, hearing, and vision differences. Individuals experience very high health disparities compared to those who don't have developmental disabilities or even any disabilities at all. But in fact, people with disabilities just get the attention of 4% of the NIH research dollars. About 10,000 roughly R01s that focused on disability were awarded over a 5-year period between 2018 and 2022. During that same period, according to the NIH data book, the total sort of dollars, or I'm sorry, the number of grants awarded was about 250,000. So that's 4%. And that doesn't really match what you see on the right side, which is a fairly high prevalence of 27% of US adults who experience a disability. That's the dark blue. And in fact, I was on a call last week, not me personally, but it was a larger call about disability research, and the director of the NIH was on there. It was one of the roundtables. And she actually said, well, if 27% of people with disabilities, I'm sorry, if 27% is the percentage of people, adults in the US population with disabilities, or are we funding studies that then have 27% of participants or an individual with a disability? And there was really no answer at that moment. So, all right, I'll let, if you could do the next slide, and I think Patrick, right, we'll do this. Could you please at that?
Thank you so very much. Hello, everyone. Yes, my name is Patrick. I'm the research coordinator for the D2R3 project. I apologize. I'm having some lighting issues in here. I apologize. I'm just going to talk a little bit about how D2R3 connects to the goals of translational science. So we know people with disabilities receive fewer health services and experience negative health outcomes at higher rates comparative to their counterparts. Some examples of these are diabetes, heart disease, and obesity, right? So people with disabilities, misrepresentation in health research directly influences health disparities in that population, right? So thinking about things like the NIH designation, right, which was designated last year, September 26th, we understand that there's a feedback loop between like researcher dynamics and the health and the disabilities community. Things like the NIH designation frames people with disabilities as a disparity population for NIH research and has a potential to address this misrepresentation in the gap through solutions such as like increased funding and targeted funding as well for disability health research. So understanding those barriers to inclusion is what the D2R3 study is focused on uncovering. And we hope that by uncovering these barriers and facilitators, we can begin to address the reasons why people with disabilities are misrepresented in health research.
Thank you. Okay, if you could do the next slide, please. Thank you. So just totally briefly, the participation opportunity is for our AIM-1, which is really, it's a survey. It'll probably take you no more than 15 minutes, which we are seeking a very wide, like a diversity of people involved in human subjects research. We're seeking study coordinators, research assistants, PIs, postdocs, et cetera. And the items pertain to knowledge, attitudes, biases, and perceptions about barriers and facilitators to including people with developmental disabilities. I'm not going to go into the rest of it just because for time. If you could please go to the next slide. And just briefly, here is the link to the QR code. I thought there was a different slide with a nicer picture. But We will also, make sure that you get a copy of this, but we are just kind of sending this out there. Again, if you have 15 minutes to spare, the only thing is if you could do it on a work computer, not a phone, it won't really work too well. And when you get to the end where you're able to choose your incentive, or to take the incentive as a gift for yourself or to donate it, you won't be able to do that if you do it from a phone. So I think if we give folks a minute, you can scan this in now. I believe you can scan it right from here. Okay. And I don't know if
We can also send this out after.
Thanks a
Thank
lot.
you. Thank you so
Thank you
much.
so much for presenting. Appreciate your time today. Good
Thank
luck.
you.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye. All right, everyone. Our speaker for today is Dr. Mike Semanik. He's a nephrologist and a physician informaticist at UW-Madison. Many of us know him well. Those of us that have tried to recruit clinical trial participants through the EMR are very grateful for his support and his expertise. And today he's going to be talking about all things relevant to recruitment as it pertains to the EMR and any other thing, Mike, that you have to share with us. So I'll turn it over to you.
All right. Thank you very much, Nazia. And thank you everyone for coming. I'm going to share my screen and get that up and start the presentation. All right. Everybody see it okay? All right. See the thumbs up. Thank you. So as Nazia was saying, I'm going to try and give a broad overview of some experience that we've had with the intersection between recruitment and technology. A lot of that technology revolves around the electronic health record, but we'll be talking about some other technologies as well. I tried hard to not talk about necessarily specific services or specific things that we're doing here at UW. I tried to keep it a little bit more big picture than that, but I am, of course, very happy to answer any questions that come up, including questions that are more specific to what we're doing here. So what I'd like to talk about today is I tried to divide it into three major categories of challenges that have come up. The first one is that there is a persistent gap between what humans are doing and saying and what technology can understand. And so we'll talk about that. And then there are some challenges that are predominantly on the technical side and some challenges that are predominantly non-technical. I originally, in the first draft of this, called them human challenges, but that didn't really seem quite fair. So I switched to saying that they're not predominantly technical and not necessarily human related. And then we'll be talking about opportunities we have to meet each challenge and hopefully overcome it with time and effort. So the human technology gap. So for my whole time doing informatics, which is almost 10 years now, there's been a very persistent gap between clinical technology or terminology on one side and then informatics terminology on the other side. And people have made a lot of efforts to bridge that gap, and we're going to talk about some of those efforts, but I would say the gap definitely persists. And to give a really simple example of that, you can say well-controlled hypertension. You can talk to most clinicians and say, well, this patient has well-controlled hypertension. They have a general idea of what that means. It doesn't necessarily involve a lot of additional explanation beyond that. But if you try and tell an informatics person that or a computer that, they're really going to have no idea what you're talking about. And to give a quick example, so if we say, for the sake of argument, we're going to say that normal blood pressure is less than 140/90. So here's an example of two patients with multiple values over a few months. Teresa, did I have to do anything to set up the poll, or are we going to do an informal poll?
I'll launch it. So I can launch it right now. 1 second.
So I was curious what people would think the answer to this, and I'll preface that I don't think there's necessarily a right answer, but I'm curious what people will think the answer. Do either of these patients have well-controlled hypertension, or do both of these patients have well-controlled hypertension?
So for people that are looking at this, what's showing up in the survey is patient C and D, but that's really choice C and choice D, as I'm sure you gathered.
Oh, I see. So we're about 40% have participated.
All right. So, is there a way for me to see the answers? Or for everybody
Yeah,
to see the
I
answers?
will. I
Okay.
will show the answers when we kind of get a little more participation, if that's okay.
Sure. Yeah, I think you can make the argument patient A had some higher blood pressures under better control recently. Patient B has lower blood pressures, but sometimes they spike up. Maybe both are controlled, maybe neither are controlled. Maybe it's just patient A because you're going off of recent values.
Teresa, you could share the results
Okay,
now.
we're at about 67% and show results.
Y'all are wonderful. This is exactly what I was hoping for. That, yeah, there isn't really a right answer here. You could make an argument for any of the answers being correct, and you all split them so nicely. That's great. So that's sort of the whole point, is that we can say well-controlled hypertension, but we don't have a good informatics definition of it. And we could try and come up with one. We could say that maybe we're going to be pretty strict and we have to have 5 out of the 6 blood pressures that are within a goal, and the patients have to have at least 6 blood pressure meds within the last 12 months, and 5 of those 6 have to be a goal. But then you get other problems, which is that, there's probably a lot of patients out there that don't check their blood pressure that frequently, or at least if they're checking it, it's not necessarily in a place that we can access it and use that data to determine whether or not their blood pressure is well controlled. So we are going to talk about missing data and interoperability, but for right now, I want to focus on this major problem, which is that these informatics terminologies can't handle nuance. And that's where this gap between the clinical terminology and the informatics terminology comes from. And so what do we do about that? How do we bridge this divide? There's really 2 broad options. The first is you can either try and get rid of the nuance, which is sort of what we were trying to do in that last slide when we were saying, well, we're just going to come up with a definition for hypertension, and that way there won't be any nuance around who has well-controlled hypertension and who doesn't. If we work hard enough, we can establish a definition that will fit. And that works sometimes. And then the other way is to sort of accept nuance and come up with better ways of processing it, which is where natural language processing comes in. So we'll talk a little bit about that. First, minimizing nuance. There's 2 ways that you can go about this. You can either attack it from the front end, so from the system that people are interacting with, which here at UW is called HealthLink. It's an instance of Epic. So you can either deal with it from the EHR front end, and or you can deal with it at the back end. So all the data that goes into HealthLink gets transferred into various databases every night, and there are ways that we can modify it or update it once it's there. From the front end side, to give a few examples of what the system does to try and reduce nuance, is that sometimes it will put limits on what you're allowed to put into the system. So for a heart rate, you can't put the letter A, for instance. It has to be a number. It has to be a positive number, and it has to be within a defined range that would sustain human life. So you can't have a heart rate of 1,000. Everyone knows that no one would be alive if you had a heart rate of 1,000. Then there's things like you can only order medications at specific frequencies. So there's a select list whenever you go to order a medication in HealthLink. You have to choose a frequency. You can't just put in whatever you want. It's not a free text field. You have to select from a list of predefined frequencies. Lab values generally have to be within specific ranges. You're almost never going to have a negative lab value, for instance, and they are tied to specific units. And then probably every clinician's favorite is that every encounter needs to be associated with a diagnosis, and there's only so many diagnoses you can choose from because the diagnoses themselves are tied to these coding systems. So again, diagnosis is not a free text field that you can put in. You have to choose from a pre-existing list, and it's a very large list, but it's still a pre-existing list. So that's some of the ways that nuance can be reduced. And then we can do things on the back end as well to further reduce nuance. But I want to make the point here that every time we do that, there's a trade-off that we lose some specificity in the data when we group things together to try and make them more portable across systems. And I wrote it here, losing nuance means losing edge cases, that there are some patients that are going to have a weird constellation of data. It's not necessarily going to fit whatever definition that you're trying to come up with. And so the easiest thing to do, and what often happens in these cases, is those patients just get dropped. They're not considered to be part of the set that you're interested in. In reality, they probably could have been part of the set, but because we're trying to set up very specific guardrails about who's included and who isn't, then we end up losing those edge cases. The diagram here shows the progression of data through Epic systems. So in HealthLink, data gets put in. It first gets extracted to a database called Clarity. It then gets extracted to another database. It gets transformed and sorted into different ways and gets put into another database called Caboodle. And from there, it can even be extracted further into other databases. The only reason I bring this up is not because I think anyone really cares about what happens on the back end of Epic, but because every time this happens, every red arrow here means that messy data is getting removed. We're losing some edge cases. So the further along this path you get, the more data you're losing. It's not a ton. I'd say probably like 95% of Clarity makes it to Caboodle. And then depending on the other database that you're going to, a good chunk of it makes it from Caboodle to those other databases. So you're not losing a lot of data, but you are using some. And so one of the other databases that you can move on to from Caboodle is we have here at UW and lots of other places have this is an OMOP database. An OMOP is a very ambitious, very large vocabulary that is designed to try and be flexible as possible to capture clinical information. That being said, it still suffers from the same problem that I was talking about, that we take our data from Caboodle and we map it to the OMOP standards. And there's going to be some ambiguous data, some weird edge cases that aren't going to get mapped and so get dropped. So the number of patients in Caboodle is not going to equal the number of patients in our OMOP database necessarily, because there's going to be some weird edge cases that aren't going to make it over there due to our mapping. But the nice thing about OMOP is that it's publicly available, open source, anyone can use it, and a lot of people do. So if you are working on a research project with a bunch of different institutions and you want to have really well-defined inclusion and exclusion criteria, one thing that you can do is say, I want to base it on these OMOP IDs. So I only want patients that meet these OMOP criteria. That way you can disperse those criteria amongst any institution that has OMOP. They should all be able to automatically query their OMOP database and pull the list of patients that meet those criteria. So there isn't a lot of translation that has to happen in each individual site. So that's the nice thing about OMOP. The other thing that I'm sure some people have heard about, and we could talk more about if you'd like, is Cosmos, which is basically, Epic takes all of the data at various Epic sites and combines it into one super large database that can then be accessed for various purposes. I would say more so than OMOP, data that's going from Caboodle to Cosmos, you're losing more of it. You definitely lose a lot more specificity going from Caboodle to Cosmos, because there's a lot of Caboodle that's, even though it's supposed to be pretty standard amongst Epic institutions, there's a lot of Caboodle that gets personalized based on the institution you're in. And all of that personalization has to go by the wayside when everything gets combined into Cosmos. So the amount of data that you can get from Cosmos is huge in terms of the number of patients. It's not so big in terms of the number of data elements that you can pull from it. And this isn't meant to be an advertisement for Epic, but just to keep everyone apprised of what's going on in that space. Epic is working on using its Cosmos data as an available pool for recruiting patients. So their idea is people will come to Epic and say, oh, we have a research study. We want to recruit patients with disease X. And then Epic will go into Cosmos and say, identify all the patients with disease X in the site that they're in. and they'll do that. And then they'll send that information to each individual site and say, hey, here's a research study for disease X. It looks like you have 100 patients that would qualify for this study potentially. Are you interested in participating? So that's sort of Epic's vision and what they're hoping to start doing in the coming year. So that is removing nuance from clinical terminology. So it involves placing limits on what clinicians can put into the chart and sort of forcing certain things to be mapped to other things so that the computer can recognize them more easily. The other option is to just accept that the nuance exists and try and teach the computer how to deal with it. And that's where natural language processing comes in. So this is not going to be a deep dive into NLP, but just a quick overview. Just so we're all on the same page, there's some very basic steps that have to happen for any NLP. You have to decide what chunk of text you're going to be using, which in the case of medical research is generally clinician notes. But this does end up becoming important because there's actually a lot of data in an EHR instance. There, like all the telephone notes, the MyChart notes, anything that the nurses are writing in nursing notes. There's a lot of different data that you can choose from, and it's often not the best idea to just say, "Give me all of the documents." You really should put some boundaries on what documents you're including in your NLP pipeline. Then you clean it up. So you get rid of any punctuation and you space out all the words so that the computer can easily understand where the words are. And then the key for all NLP is this concept of entities. So the computer needs to recognize individual words or phrases as entities. It needs to assign properties to those entities, and it needs to assign relationships between those entities. So really simple example, this sentence here, the patient's biopsy showed no evidence of malignancy. So there's three entities in this. There's the patient, there's the biopsy, and then there's malignancy. You can see that an NLP system that's working well would appropriately negate malignancy and would not negate patient and biopsy. And then the patient has a biopsy, so there's a relationship there, and the patient may or may not have malignancy. In this case, it says not a malignancy, so there's a relationship there. So that's how the process should work. And then we can ask the question, does this patient have cancer? And based on what we know so far, the computer would actually tell us no. Or the computer should say, I have no idea, actually, if the patient has cancer. And that's because the computer doesn't know until we tell it or until it learns in some way that malignancy is the same thing as cancer in this context, and same thing as tumor, that all of these words can be synonyms in this context. But the computer doesn't necessarily know that unless it has a source of truth that tells it that. And so the two major sources of truth, historically, what I trained in a decade ago, is that the source of truth was annotated data sets. It was basically humans going in, looking at these data sets, going through clinical notes, and telling the computer what was correct. And then you would use those data sets to train models that would then go on to predict what was correct in data that the computer hadn't seen yet. So that is still very much that whole model is very much still widely used today. And we actually have a third-party vendor here at UW that we are using for some NLP that works in that way. It is not a large language model like ChatGPT. So those are, as everyone knows, relatively new. There are a lot of similarities. I think them, and there's a fair number of differences, but I think the main point is that the source of truth for an LLM isn't someone specifically going in and annotating a document and saying this is what the computer should do. It's just that there are so many documents that are used to train this LLM that essentially the computer gets a sense of what's correct and what's not because it just sees it a million times and determines, well, this relationship happened a million times, so it's almost certainly going to be the correct relationship when I see it the millionth and first time. And of course, as we know, these LLMs are always evolving and they do rely on human feedback for improvement. So this idea of annotation hasn't completely gone away. It's just that it's not as necessary to get things trained and off the ground. There's certainly companies that are interested in using LLMs for the purposes of clinical trial recruitment. We are keeping our eyes and ears open and have talked to a few companies about that here. I have no doubt that we'll be doing this in 5 years. It's just a question of getting the resources in line and finding the right partner to work with. So that was a quick overview of the gaps between clinical terminology and informatics terminology. Before I move on to other challenges, I wanted to see if there were any questions around that.
Mike, it looks like there's a couple of questions in the chat. I
Oh,
think
okay, great.
partially address this, but yeah, could you take a look at the chat and see if you want anything to add?
So the losses when we're talking about going from one database to another, what's responsible for those losses? It comes down to you have to, as you get to these higher level databases, so Caboodle is one example, but then OMOP and Cosmos are even higher levels. They have certain rules about data type and data quality that have to be met in order to be included, that are stricter than the rules for the database below it. So Clarity doesn't have many rules at all, actually. Like you can, anything that can be put in the health link basically comes out in Clarity. But then in order to use it, Caboodle puts additional rules on top of it. So Caboodle, for instance, will want to group all of the lab values that a patient has together. And there are some lab values that come out as error or no value, or there are various strings that mean, oh, actually, this lab didn't get processed correctly. And rather than just include those free text values in there that we know are not going to be useful, Caboodle has requirements where it says you can't have those. And so those labs just get dropped out because nobody can use them anyway, and they're just taking up space. They still exist in Clarity if for some reason we really need to go back and look at them, but Caboodle is supposed to be a more user-friendly, useful version of that data, and so it doesn't make it there. So it's situations like that for the most part. And then when it's talking about going to something like OMOP, OMOP tries to be very extensive and in a lot of ways is a very extensive, but there's still things that come up where when we're trying to map from one concept in Caboodle to another concept in OMOP, there just isn't a clear mapping there. And when that happens, we just kind of shrug and say, well, I guess this data point's not making it because there's nothing for us to map it to. And so that's another reason that things get dropped out. Has there been any work characterizing the type of cases that get dropped? You know, there's a lot of work done on the infrastructure side at UW Health that runs this and comes up with these requirements for Caboodle. And I know Epic, I'm sure Epic has put a lot of work into it. I'm not aware of anything that's been published necessarily, though. It's certainly something I can look more into. Thank you for those questions.
Thanks, Mike. Those are all the ones I see.
All right. So we'll jump into some technological challenges. And this one does have a right answer, but it's more of a, I'm curious what everyone's opinion is. So I'm speaking about from a UW-Madison standpoint now. We have a service that we call CURDS that's job is to pull data and deliver that data to researchers. And so when CURDS is doing that, what's the greatest challenge that we face when we're doing that? Is it the data isn't accurate, it's not complete, it's not timely, which means that it takes a long time for something to make it from HealthLink into our systems, or is it that we have poor data governance?
we're at about 60% complete.
All right.
And we'll share the poll.
Yeah, and I would agree that the answer is B. Most of the problems, we'll go into each of these in more detail, but most of the problems we have is that people want data that we just don't have access to for various reasons. So jumping in. So data accuracy. I don't want to concern anyone about our data accuracy. It is in general very good. But the garbage in, garbage out rule applies. So if you put something in HealthLink that doesn't make any sense, we will take that same thing that you put into HealthLink and give it right back to you, and it still won't make any sense. Which is to say, we don't have additional guardrails beyond what the electronic health record provides. So the electronic health record prevents you from putting in a systolic blood pressure of 100, but it doesn't prevent you from putting in a systolic blood pressure of 20, which is probably never really an actual correct answer. But if someone does put in, say they meant to put in 200, they put in 20 by accident, the system's going to accept that. It's going to make it into the database. The database is going to say, yep, that's an integer, and it's just going to propagate that through all of the systems that we have. So there are errors that make it into these systems that obviously everybody makes mistakes, but in general, the clinicians are really good and very careful about what they're putting in there. So the vast majority of the data that comes out of our systems, I feel, is very accurate. And so the opportunity here, how do we address this challenge? And it just involves good communication between the data pool team, the team that's pooling the data, and the study team. Because a lot of times the team that's pulling the data doesn't necessarily have the clinical knowledge to say, oh, this looks really weird. Like, why is this data point here? Like, why is this patient's creatinine 100? That shouldn't even be a possible number. The data pool team isn't necessarily going to be able to know that, but the study team generally does. And so it involves some review on the part of the study team, and then we can go back and we can put additional guardrails into our pool and say like, oh, actually, I only care about systolic blood pressures that are between 60 and 200, and anything else I'm going to assume is an outlier. So there's not a great way to, we can't prevent all errors in data, but we can certainly want to keep the lines of communication open so that when errors are discovered, we can work on ways to prevent delivering that data. So data completeness, or why don't we have what you're looking for? There's two major answers to that. The first is that it just can't be discretely captured. And although we have some ability to do NLP, it's still pretty limited at this point, so we don't do a lot with notes yet. We will as time goes on, but right now we're still not doing a lot. So generally speaking, if it's not discretely captured, if it's not something you enter in like a lab or a vital sign or something like that, then we may not have a good way of accessing it. The other big problem we have is that it is captured, it's just in someone else's system that we don't have access to. And so that brings up the problem of the lack of interoperability. And so the opportunity here is to increase interoperability because people have been working on this for decades and they've come up with some pretty good solutions. The nice thing is we have the infrastructure to do it. We have these protocols that are pretty universally accepted at this point, like FHIR and HL7, that allow for easy communication of patient data back and forth between two systems. It's just a question of getting the system that you want approved to talk to our system. So it really becomes more of a policy protocol problem as opposed to a technical problem at that point. Data timeliness. I'm going to do a quick segue here. So our data is updated every night, and I think pretty much every major hospital center does this the same way, where they take all the data that was entered into their EHR and they pull it every night, extract it to their databases, so it's available for people to use the next day. So nothing's like real time by the minute, but everything should be pretty up to date. It's all no more than 24 hours old. But I think one of the things that we are going to be focused on here, and I think other people around the country are focused on, is how can we reduce that lag even further, and do what we're terming more real-time recruitment. So if this is the current process where, if things get entered into HealthLink and then it gets extracted to the database, our team pulls it from the database and sends a report back to the people that requested it. This process is taking days. We'll measure that time in days. What we're hoping for, a nice future state, would be something like this, where things are getting put into HealthLink, and rather than worrying about it, waiting for it to get extracted to the database, we're going to have a research computing platform that's going to be automatically pulling some data as it gets entered. It's going to go into our research computing platform. That platform is going to be able to talk to the database, so it can pull older stuff if it needs to, and it's going to process in real time and say, hey, is this patient going to be a good fit for the study based on the data that we have in the database here, perhaps some other databases over on this side, um, and what just got entered into HealthLink. It'll combine all of those things. And then rather than us sending a separate report, it'll bring it back to the person who's putting in the original data. It'll pop up, if not in HealthLink, at least on the same computer, and say, hey, this patient might be a good fit for this study. So this is all going to be measured in minutes as opposed to days, and hopefully involve a lot less waiting and fewer systems where you're not having to go to access a different system to get your report of potentially eligible patients. It'll all be actionable while you're sitting there at the computer. This is a dream. It's a goal, and it's something we're going to be working towards. I don't know how long it will take to get to this point. I said earlier I was confident we're going to be using LLMs in 5 years. I'm less confident that we'll be doing something like this in 5 years, but it is definitely a long-term goal. And then data governance. So the only point I wanted to make here is that the way data is captured in the electronic health record and the way it's structured in the databases that we use is pretty much all set up for clinical care, which is 100% appropriate. But it does mean that sometimes we have to pull things from weird places, and it's not necessarily as straightforward for research needs as it is for clinical needs. And so I don't know that there's a lot of opportunity for individual study teams to do much with this, but we do have chats with Epic and about developing their research module further and how they can get more data into Epic so that we can pull it on the back end and make it easier for people to identify patients and also just track the patients that they already have on their studies. So I think that anyone who's used Epic's research module hopefully feels that there have been some improvements made over the year, and we will continue to advocate for more improvements along with other institutions from around the country. I will check the chat real quick, make sure there's not more questions. All right, any questions about those things before I move on to this last section?
Please unmute yourself for your question, or you can post it in chat too if you like. Mike, one question that has come up is the extent to which one can extract data from different entities, like, places that we partner with, for instance, the rehab hospital or Northern Illinois, and what's allowable and what's not. So maybe you could speak to that a little bit.
Sure, yeah. So I'll group it into two big categories. The first category is care everywhere data. So data that you can see in HealthLink, but it isn't from any UW institution or any kind of joint venture. So UW has no involvement with it. That data is not available for research, and it's actually really hard to get to. It's stored in a completely different spot in the system. So by default, our teams don't have access to it. So even if it was allowable for research, we wouldn't be able to pull it because we don't have access to it. So all the care everywhere data, unfortunately, does not make it. The joint venture data it does make it into our systems. And so it is technically possible for us to pull it. And then it becomes a question of, is it allowable for us to pull it? And that really depends a lot on the venture. So Northern Illinois, it's usually pretty straightforward to be able to use that data. We need to, for retrospective data, we basically just need to tell Northern Illinois what we're doing. They like to have a list of pools that we do for them, but they have allowed us to use their retrospective data for retrospective pools. For recruitment, it and this hasn't come up a lot just because the clinical trial infrastructure in Northern Illinois is less developed than it is here in Madison. But it is theoretically possible to do that. You would just need additional approval from Northern Illinois' IRB to do recruitment there. So with that approval, assuming you have personnel in place down there that could help you, we can certainly provide Northern Illinois data. For some of the other joint ventures like Generations or Wisconsin Sleep Center, um, again, the retrospective data isn't as big a deal, but for recruitment, we're not allowed to send out data about those patients. Take a step back. Patients can't be recruited from those places. They have to, their recruitment has to come in some other way. So there was a study recently, for instance, that was looking at people who were going to be discharged from the rehab hospital, and they couldn't be recruited while they were there at the rehab hospital because of the legal agreements that we have with them. But once they were discharged, they could be recruited. So we can use their data to identify them. They just can't be recruited while they're in certain spaces, if that makes sense.
Yes, thank you. That was very helpful. Okay, not seeing other questions, so you can go ahead, Mike.
All right, I'll finish with a few non-technological challenges. So the first involves workflows. So I just wanted to tell a quick story about MyChart recruitment, which we've been using here at UW for about a year and a half now. It's gone well overall. We're excited about it. We're certainly happy to help you use MyChart for your study if we can. Overall rates, and this I'll say this varies widely by study, but the average overall response rate for MyChart recruitment is about 25%, which is pretty good. And I think one of the reasons it's pretty good is because we've made it really easy for participants to respond to it. What happens is they get a tickler that says, "Hey, you have a MyChart recruitment request," and they can log into MyChart and they can look at it. And they're looking at it right there. They can either say, "Oh, I'm interested about this," or they can say, "No, thank you." And so they just click a button, and that's their response. The other thing that's nice about it is that it really allows study teams to have more control about how many invites are going out and the timing of when they're going out. So we've had a couple of studies where they want to send it out like a week before some event happens, like a week before they're going to be seen in clinic. So they get the invites sent out. It'll go out a week before they're seen in clinic, or a week to I guess it could be exactly a week before they're seen in clinic because it's dependent on the study team. And then that way, hopefully they've had time to look at it. And during the clinic appointment, they can bring it up and say, "Oh, hey, we wanted to tell you more about the study. Did you get our MyChart invitation? Is it something you're interested in?" So that's been a really nice perk of it too. But we also had some issues with some studies that were good for learning. And so that's one of the things I wanted to bring up today. So from the study team perspective, this is the general workflow, is that we will deliver a list of patients within HealthLink. You can see this list here. The study team will then go into the individual charts and confirm that the patient meets their eligibility criteria. And if they do, then they'll hit this button and send them a recruitment request. So we thought this is great. It's very straightforward. It all happens within HealthLink, which is nice. But really, all of these workflows were designed for the kind of study where they're sending recruitment either once or in batches. We weren't really set up for a kind of study where people would be eligible for it on a recurring basis. And because of that, we didn't think a lot about how patients got off that list. So patients get on this list by my team identifies that they could be a potential fit, and they get a flag put on their chart, and then they show up on this list. But we didn't think as much about how to get people off the list. If they get sent an invite, they'll fall off that list because that flag changes from identified to invited. But as we were rolling this out, we realized that there were some cases where people were ineligible for the study based on whatever criteria at the moment, but the study teams didn't actually want to fall off the list. And so they would just sit on the list, and then they could be revisited in the future and see, okay, well, maybe they're eligible now, even though they weren't before. And so for, a list of 100 patients that we're sending one time, that's not a big deal. But when we got a study that was a list of 100 patients that we were sending once a week, it became really problematic that patients were not getting removed and that we didn't really have this straightforward way of removing them if they weren't invited. So the list just got longer and longer and longer until it was 10,000 people long, and it was totally unmanageable. So that led to a lot of opportunities for improvement. We refined the inclusion criteria a bit so that we weren't necessarily delivering 100 patients a week. And then we had to come up with a whole new workflow around how best to remove patients that we didn't want to be permanently marked as ineligible, but weren't eligible at the moment. And so we came up with a new workflow around that. And then, of course, there was always the option to say, well, maybe my chart recruitment wasn't the best in this case, and is there any other methods that we can use to help you? So the lesson there is that workflows matter. And there wasn't necessarily anything technical. It's just that we didn't necessarily anticipate this particular case. And so we learned a lot from it. And then the other challenge that I'm sure everyone on this call is very familiar with is this concept of external validity, where we want the clinical trial population to be representative of the broader population of interest. And so if you have disease X and disease X has the population has various characteristics that you can see on the left here, and they break down into 3 roughly equal groups. But then in your study population, you're only getting this yellow chunk. You could see why people would question, well, maybe the results of the study are helpful for determining what to do with this yellow chunk, but maybe it's not as helpful for the red chunk or the blue chunk. And so you really want to do everything you can to have these 2 pie charts to look as identical as possible. And that's hard because there are a lot of different factors that you want to balance, more than even I listed here. And it's also hard because some of these factors aren't necessarily easily defined in the data that's available. I'm looking at socioeconomic status as a big one. We just don't really have good data about that in the electronic health record. The other thing to consider is that we have a lot of different recruitment methods, but each of them have their own potential biases. So these are our sort of big 4 recruitment methods that we work with. We do phone calls, mailings, MyChart, and then in-person recruitment in clinic. But all of them have their shortcomings, and I wanted to highlight that MyChart specifically, we've done a lot of work looking at the MyChart population versus the general UW Health population, and there definitely is a slight bias there in terms of people who use MyChart are more likely to be in an urban area, more white, and younger in general. Not necessarily, there's still a lot of elderly people that do have active MyChart accounts, but it does skew younger for sure. And so these are all things to take into account when you're working on your recruitment plan and trying to determine how are we going to be able to reach people. Generally, in-person recruitment in clinic I think it just works the best. It's not always feasible, I understand that, but certainly when it is feasible, I think it works really well. And then usually what we recommend is a combination of different routes so that we can try and minimize the bias that any one route provides. And then there's a lot of back and forth between the recruitment team and the study team to help develop the recruitment plan. And one of the things that we like to provide is we can provide you any of the demographic data that you would need to help balance that study population in a way that you want. So if you're looking for race data, we can provide that. If you're looking for sex, we can make sure it's balanced by sex. We've had a few studies recently where he said, I want to make sure that we invite the same number of men and women. And so we'll just randomly select 100 men and randomly select 100 women and say, here's your data set, and you can invite all of them. So we can do stuff like that. We are able to use the area deprivation index, which is developed here at UW-Madison as a way of taking census tract information and trying to connect that with socioeconomic information. So it gives a rough proxy of socioeconomic status based on the patient's address. We can send out ADI scores with our data sets when that's of interest to study teams to help balance things that way as well. So in summary, a lot of the challenges that we face has to do with this nuance that exists on the clinical terminology side that doesn't necessarily translate well to informatics terminology, the two ways to deal with that is either to try and force the nuance out by restricting people's options or NLP. There's technical challenges such as interoperability, which we're working on overcoming, requires good partnership with the study teams, and also just the available resources to be able to connect different systems together. And then there will always be non-technical challenges like workflow development and achieving external validity that we recognize are issues and we want to help study teams however we can. So whether that's a consult to determine the best method of recruitment or additional demographic data that study teams need to try and balance their recruitment profile, we're happy to provide that. And I saw a few things pop up in the chat, which I'll look at, and I'm happy to take any more questions.
Thanks so much, Mike. That was fantastic. Yes, it looks like there are 3 questions in the chat. And others, if you have questions, please put them in there. So the first one is, do we have an update on when MyChart recruitment can be used for pediatric studies?
Yeah, great question. So for the MyChart for pediatrics is broken into 2 groups. There's the 12 to 17-year-olds and the under 12-year-olds. For under 12, we piloted it successfully this last month. And so we are saying that for under 12 pediatric studies, MyChart is now open. The 12 to 17-year-olds have additional legal considerations given their adolescentness. And so we're piloting that in the coming months here in January, February. So I'm hopeful that will go well, and that by the spring or certainly the summer at the latest, we'll be able to have MyChart recruitment for basically everybody.
Thank you. Have you gotten any patient complaints to MyChart recruitment messages? Is a question.
We track so the IRB has required, and we agree with this requirement, that you provide information to opt out of MyChart recruitment on any MyChart recruitment message. So any study team that's worked with this knows that there's a template you have to fill out, and the template has if you want to opt out of receiving these messages, call this number. And so we track how many people call the number and opt out, and it's not many. I think it's maybe up to 9 now, and we've been doing this for a year and a half. So it's a really, really small percentage. And we don't really provide another venue for them to complain. So I'm not sure if there's frustration out there that we're not capturing, but I certainly haven't heard any. And as I said, people haven't been going through the trouble of opting out now en masse.
Thank you. How do we get started on using the MyChart recruitment option? Is there a link or a point of contact?
Oh yeah. Well, you can email me, or I can. I'll find our consult request form, and I'll throw that in the chat.
That would be great. Other questions from people in the audience? Feel free to just chime up if you have something you'd like to ask. And the link is in there. So Maria, hopefully that answers your question. Okay. Last call for questions. All right. I'm not seeing oh, there's one. Can you use MyChart recruitment if your appointment is outside the HCC?
Yeah, that is a great question. The short answer is maybe. but you have to, so your study has to be inside the HCC. You have to have a partner within the HCC. And that partner within the HCC has to provide the staff that are able to do it. Because MyChart Recruitment all the workflows are within HealthLink, you need to have study staff that have HealthLink access. And so and there's been a few studies where, for instance, people out in kinesiology have been working with oncology because they're looking at cancer survivors. And those studies have been able to use MyChart Recruitment because the oncology staff has agreed that they're going to take on the responsibility of sending out those MyChart messages and then responding to anybody who replies to it. So as long as you're able to partner with somebody and they have the staff that could do it, then yes. But it does require that partnership, and it is a decent amount of work for the people inside the HCC since they're taking on the bulk of their recruitment duties there.
What about if someone doesn't have, is in part of the HCC but doesn't have UW Health access? For example, a non-clinician, I guess, would be one example. So
So,
yeah.
yeah. There's someone that we're working with in the School of Nursing. You can get access for the purposes of sending out HealthLink messages. Study coordinators sort of get that access by default as part of their role, but PIs can take a training and then submit a CSAR request to get the necessary functionality to be able to send out messages. So that is possible if you're inside the HCC but don't necessarily have a study coordinator that can access HealthLink. The PIs can sometimes do that themselves.
Great. Well, we are at time. Again, thank you so much. Much appreciated. And we will share the recording of this talk as well. Thank you, Mike.
Beth, just shoot me an email, and there's not really a website. It's it's unfortunately not as straightforward as that, but I'm happy to email and talk to you about it.
All right. Thank you, everyone. Have a great rest of your